Hi guys, so today we're going to be talking about cells. So the first topic we're going to go over is classification, like types of cells, and then surface area to volume ratio, and then the phospholipid bilayer, and then cell transport will actually be a part of a different separate video, and then we'll be going over organelles. So we're going to start off with the basic 3 domains of life. So it all started about 3.5 billion years ago. Bacteria and eukarya split off, and then archaea split off from eukarya later on. And so those are 3 domains. And then something still interesting, in the endosymbiont theory, it is said that mitochondria and chloroplasts arose from bacteria and were consumed by eukarya, and this is actually proven through such evidence as the double membranes of both the organelles and the fact that they both have their own DNA. So that's just a fun fact. Moving on to compare the 3 domains of life. Bacteria and archaea are both prokaryotes, which means that they are single-celled and they do not have a membrane-bound nucleus or membrane-bound organelles, whereas eukarya are eukaryotes and therefore are single or multicellular, and they have membrane-bound nucleus and organelles, which allows for compartmentalization and more specialization within the cell. Now, at this point, bacteria and archaea may seem very similar, and up until recently, archaea were actually considered a subset of bacteria known as archaeobacteria. However, we now know that that is not true, and as you can see here, they are actually quite different. So one of the main reasons is evolutionarily they're different, as seen in the earlier diagram. Bacteria and eukarya branched off, and then archaea branched off from eukarya later. So that's one of the reasons. Moreover, the RNA polymerase in archaea is more complex, and RNA polymerase is basically just an enzyme that catalyzes the synthesis of RNA and transcription, which is a part of gene expression, and we'll probably go over that in a later video. And then one of the other reasons is the cell wall, and so in archaea, the cell wall does not contain peptidoglycan, whereas it does in bacteria. So moving on to the second topic, which is surface area to volume ratio, we can start off with a quick example. So here we have a 1 inch by 1 inch by 1 inch cube, and the surface area is 6 inches squared, and the volume is 1 inch cubed. And so the ratio turns out to be a 6:1 ratio. And then here we can see in our second example, this is a 3 by 3 by 3 cube. The surface area is 54 inches squared, while the volume is 27 inches cubed, and so that comes out to a 54:27 ratio, which should simplify to simply a 2:1 ratio. And so now if we compare the 2 surface area to volume ratios, we see that the 1 inch cube has a much larger surface area in comparison to volume. And so that's something we should keep in mind as we move on to see how surface area and volume affect cells. So cells are extremely small, but as humans grow, these cells don't grow with us. Rather, they divide, and we end up getting more cells. And this is because cells have a size limit, and this is due to the surface area to volume ratio. It's ideal for a cell to have a larger surface area as compared to its volume. So if we compare it to our earlier 2 examples, it would be ideal for a cell to be the size of that 1 inch cube as opposed to the 3 inch cube, because there's a higher surface area as opposed to volume. Now, this is because a larger surface area allows for more exchanges between cells and other cells or cells and the environment, and it allows for more nutrient intake, which is what allows for a cell to survive. And now on to our third topic, the phospholipid bilayer, or the cell's plasma membrane. So this cell membrane is made up of phospholipids. Phospholipids consist of a phosphate head, glycerol, and 2 fatty acid tails. And this phosphate head region is hydrophilic, which basically means that it is able to mix with or absorb water, whereas the fatty acid tails are hydrophobic, which means that they repel water. So here we can see that the phospholipid bilayer is basically that outer part of the cell, that plasma membrane of the cell, and it's made up of a bunch of phospholipids, and it's kind of like a phospholipid sandwich with the heads on the outside and the tails on the inside. So here we see basically that the outsides of the membrane are hydrophilic. So the parts facing the outside of the cell and the inside of the cell are both hydrophilic, whereas the inside of the membrane, which is not facing anywhere outside, the inside part of it, that is going to be hydrophobic because of the fatty acid tails. Now this is a section of cell membrane with just some of the main components. This is a protein channel or transport protein through which large molecules or polar molecules can pass through, because the hydrophobic inside of the cell will not allow polar molecules to pass through, and the nature of the plasma membrane won't allow large molecules to simply diffuse through, and so there will be a lot of proteins, different types of transport proteins actually, that will allow molecules to pass through the membrane. This is a carbon chain, which basically serves as an identifier or a marker for different special types of cells. And these are cholesterol, which are saturated lipids that act as stabilizers for the cell membrane, especially against temperature, because in lower temperatures the cell membrane becomes less fluid, and in higher temperatures the cell membrane ends up becoming extremely fluid, and so high amounts of cholesterol allow for lower fluidity, and low amounts of cholesterol allow for higher fluidity, which can either way help stabilize the cell membrane fluidity. For the fourth section, cell transport and osmosis are a really big part of cells and how they function, but I thought that this was actually too big a topic to put into this video, and so I'm going to make a separate video, which I'll make sure to link down in the description below. And now, last but not least, organelles. I'm going to be showing organelles on both plant and animal cells and do a sort of side-by-side to show some of the differences, but also a lot of the similarities. Also, sort of to connect what we did in our first section of the video, animal and plant cells are classified as eukaryotes. And so this first organelle, some may argue the most vital organelle, is the nucleus. The nucleus contains the DNA, the hereditary genetic material, and it also coordinates the cell's activities and the function of the cell. It is that control center of the cell. Also contained inside the nucleus is the nucleolus, which houses the ribosomal RNA and basically is a ribosome factory. And we'll talk about ribosomes later on when we go to discuss the ER. So moving on, right next to the nucleus you can see the rough ER, and then that green blob is the smooth ER. And these two make up the endoplasmic reticulum. So the rough ER is called the rough ER because it's studded with ribosomes, which are needed for the translation process of protein synthesis, which we'll discuss in a later video. And the rough ER is basically the protein factory. It's involved in protein production. whereas the smooth ER is involved in lipid production, and the smooth ER does not have any ribosomes. The smooth ER is also involved in detoxification, and it stores calcium. This purple stack is known as the Golgi apparatus or Golgi bodies, and they're basically sort of a post office, so they work to modify, process, and store lipids and proteins, and they also ship off these products that they modify using vesicles, and they'll be shipped around anywhere that's needed in the cell, or they may be put in a vesicle and shipped outside of the cell. Next in red is the mitochondria, famously known as the powerhouse of the cell, which basically just means that it is the site of cellular respiration, which allows for the production of ATP or energy, which is needed for all the different functions and processes involved in a cell. Next is the chloroplast, which unfortunately is not shown in the diagram, and this is only found in plant cells, so it is not in animal cells. It has chlorophyll pigment, which accounts for the green color found in plants, and this is the site of photosynthesis. So this is where light energy is used to synthesize glucose, which can then fuel the other activities of the cell. And the purple blobs shown here represent vacuoles, which serve as storage centers for things like water, food, and waste. And in plants, there's something called a central vacuole, and so as you can see in the diagram, the vacuole in the plant is much larger than it is in the animal. And this black circle here represents a lysosome, which is an organelle that contains enzymes that work to break down materials both from outside and inside the cell. So for example, it could possibly break down an old organelle that was from inside the cell, and it works as a sort of digestive or recycling center. The blue circle represents a peroxisome. The main function of this organelle is to carry out oxidation reactions that yield H2O2 or hydrogen peroxide. Enzymes such as catalase later break down the H2O2 into H2O or water. A common oxidation reaction that would take place in this organelle is the oxidation of fatty acids, which allows for the release of metabolic energy. The two black rectangles on the animal cell represent two cylinders made up of microtubules, individually known as centrioles, or known as a unit as centrosomes. And these organelles are only found in animal cells, not in plant cells, and their main function comes up during cell division, at which point they are replicated, and what they do is they produce microtubules that attach to the chromosomes and allow them to split during cell division. As these organelles are not found in plant cells, it is clear that they're not needed in cell division, but it is suspected that they decrease division errors. And that's all for this video. Thank you so much for watching, and if you found it helpful, don't forget to subscribe.